Hello everyone, welcome to our interviews for the South American Mycorrhizal Research Network. So today I'm with Carlos Rodriguez-Ramos.
Hi Camille, thanks for having me. I'm excited to share this paper with you. It's my first authored publication, and that's always very exciting. So appreciate you being interested in it.
Excellent, congratulations. The name of the publication is Changes in Soil Fungal Community Composition Depend on Functional Group and Forest Disturbance Type. And it's actually impressive in new phytologists. So you are originally from Puerto Rico, where you did your bachelor, and now you are a PhD student at the University of Alberta in Canada.
Yeah, that's correct.
And so I understand that this publication is part of your PhD thesis.
Yep.
So what can you tell us about it?
Yeah, so it's pretty much the first chapter of my PhD thesis, and the main idea came from kind of like an overall ecological interest in a sort of like what's the structure and composition of fungal communities in our local forest here, since we know that disturbances change the assemblages of communities, not only fungal communities, but pretty much all biotic communities, by resetting the ecological succession of the system. And now with the effects of a changing climate, we're already seeing sort of like an increase in the disturbance regimes experienced by these systems. And this includes, frequency and intensity. And for example, something that is happening pretty much worldwide, but also here in Western Canada, it's increasing the intensities of the wildfires. We saw the wildfires in Australia, but same here in our local forest. We're having wildfires that are like way more intense than they used to be. And on top of this, because of an overall warming climate, we are also having in our forest of Western Canada the expansion of this bark beetle insect that attacks one of the dominant pine species in the area, which is lodgepole pine. Mm-hmm. So this little insect that it's like 5 millimeters have attacked like millions and millions of hectares of trees, resulting in like mass mortality of these trees. So that's like big part of what motivated this study to seeing what's the response of fungal communities to these different disturbances, and especially because there's the possibility of multiple of these disturbances interacting, creating like cumulative stressors that could that are like higher than what these systems have previously been exposed to. So I wanted to know what's the response right now, and seeing like what would be the response in the future as well. So, yeah.
Great. Yeah, that's a lot of factors. So how did you design your experiments in order to test all these different factors?
Yeah, so we pretty much scouted for forest, and we found for like intact lodgepole pine forests that we sampled the soil from. And for each of those intact forests, we also had a paired disturbed site. So we had sites that were impacted by either a wildfire or mountain pine beetle outbreak, which is the beetle that I talked about earlier, or that were clear-cut logged or harvested. And other forests that were also salvage logged, meaning that they were previously attacked by the mountain pine beetle, and they were following the attack of mountain pine beetle, they were logged. So that's one of the cumulative disturbances. Mm-hmm. And we know there's been some studies already looking at individual disturbances and what's perhaps the response of multiple guilds to that one disturbance, but we wanted to test whether the different filters that the different disturbances impose, let's say the heating associated with fire or the long-term decomposition and mortality associated with a bark beetle insect attack, or the sudden removal and mortality of the overstory associated with logging. We wanted to test whether these different filters resulted in a similar assemblage or different assemblages of a soil fungi. Not only ectomycorrhizal fungi, which are the ones associated with the pines, but also arbuscular mycorrhizal fungi, saprotrophics, fungi, and pathogenic fungi, because we know that these guilds interact with each other. So the response of one can depend on the response of the other. We sampled the soil in each of these forests, intact and disturbed forests, and extracted the DNA and sequenced 2 different genetic markers, one for ectomycorrhizal, saprotrophic, and pathogenic fungi, and another one for the arbuscular mycorrhizal fungi. And we also extracted and quantified ergosterol, which is kind of like a measure of fungal biomass.
And so what did you find? What happened with all these disturbances? So you mainly looked how the different fungal guilds responded to the disturbances.
Yeah, so seeing whether, and I'll use the term abundance, even though that we know that using reads and sequencing, it's not necessarily abundance, but whether they became more or less abundant after each disturbance, their diversity increased or decreased, and also how is this associated or how it relates to the soil organic layer disruption that happens after disturbances, because we know that some disturbances either compact, remove, or combust the soil organic layer, and those are wildfire and the logging disturbances. We know that mountain pine beetle being a biotic disturbance, it doesn't affect the organic horizon because it just leaves it intact.
And so in general, the ectomycorrhizal fungi tend to decrease with disturbance, and the saprobes and the arbuscular mycorrhizal fungi tend to increase, right? So Is it a good thing? Is it a bad thing? What do you say?
I don't think it's for me to say whether it's a good thing or a bad thing. It for sure it's part of that restarting of the ecological succession. I think what would be bad is if, like now with this cumulative disturbances or multiple disturbances imposing cumulative stressors on the system, whether that keeps making more and more effects. Like if the ectomycorrhizal fungi keeps declining while saprophytic keeps increasing, as well as arbuscular mycorrhizal keeps increasing, and that could change the dynamics of the forest if this keeps happening in shorter time periods. So yeah, with those more frequent disturbances, that it could be affected.
So there is this phenomena called Gadgil effect, who basically means that if you have a decrease in ectomycorrhizal fungi, let's say abundance or activity, then the competition with saprobes is less important. And so the degradation of the organic matter tends to increase because there is less pressure on the saprophytic fungi. So do you think that this is basically what is happening in these systems?
Yeah, for sure, part of it. We see in one of our figures, we have the relationship between the depth of the organic layer and the frequency of ectomycorrhizal fungi. And we see that with more disturbance of the soil, we lose more ectomycorrhizal fungi. And we also see that saprophytic fungi, it's increasing after these disturbances. And yeah, definitely there's some sort of competition in the organic layer between ectomycorrhizas and saprophytic fungi, where ectomycorrhizal fungi kind of like mines. It has an organic nutrient economy, so it lives in the organic layer and mines that organic layer for nitrogen and prevents saprophytic fungi from being active in that layer. So it results in the accumulation of soil carbon and soil organic layer, right? And then when we have a shift in dominance from ectomycorrhizal to saprophytic fungi, we see how is there more activity from the saprophytic fungi, and thus there's more turnover of that organic matter, which results in loss of soil carbon in that organic, and specifically in that organic layer. So yeah, there's that competition. And then there's also the arbuscular mycorrhizal fungi that together with the saprophytic fungi, they have more of an inorganic nutrient economy. Well, arbuscular mycorrhizal fungi depends on the activity of saprophytic fungi for their nutrient economy. So yeah, we see how the increase in saprophytic abundance can benefit the abundance of arbuscular mycorrhizal fungi in the soil.
Yeah.
Yeah.
It also depends a little bit of changes in vegetation. I was thinking you talk about a lot about fire, right? When you have a fire in the forest, then probably the vegetation can change quite a lot, and this can have a big influence on the type of mycorrhizal fungi that you will find below ground.
Yeah, absolutely. That's one of the reasons why in intact forest, arbuscular mycorrhizal fungi was so abundant, because these forests are dominated by pine species that only associate with ectomycorrhizal fungi, and that creates these positive feedbacks, plant soil feedbacks that increase the abundance of ectomycorrhizas. But then when we lose the overstory composition of the forest, meaning that we lose all those pines due to the disturbances, specifically, like mostly wildfire and the logging disturbances that result in the complete removal of all the pines or combustion of the pines, then we would have other plant species, mostly from the understory and ground cover, that would start colonizing the soil, and those plant species likely associate more with arbuscular mycorrhizal fungi. So that's part of like what's increasing or like aiding the increase of arbuscular mycorrhizas in the soil, for sure.
Mm-hmm. And so finally, because there are so many factors, no, and they are all interacting together.
Yeah.
And so today, especially the studies looking at like global change or carbon storage, people are trying to do some kind of large-scale prediction. Do you think there are some of these factors that somehow have more weight than others and that we could use as predictions? Or you think it's impossible to read?
Yeah, it's as you say, it's like a lot of factors coming into play, and it's hard to untangle the individual contribution of all these factors. And part of it, would be sort of like what's the system we're looking at. I think it's hard to make a model for the whole global system. Because we know that in my case, I work more in boreal systems. We know that the ecology and the dynamics of these forests are different from those in the tropical, in a like tropical rainforest, right? So yeah, I think it's really hard to say this is what's causing the major changes in the system, but definitely disturbances are something that are happening worldwide, but there are also different disturbances, right? Here in coniferous forest, we have lots of wildfires and insect outbreaks. In the tropics, we have lots of hurricanes and like droughts and others. So yeah, I think each individual disturbance results in specific changes in the system that have to be considered in part individually, just so we can understand like what's happening in that system, as well as how can we remediate that system specifically, rather than make enough like a whole like guideline for everything. So yeah, I think it's quite hard to untangle every single factor and and try to make a global prediction of it. But
so is it where you're working right now? I understand that you're about to finish your PhD, right? So
yeah.
what are your research interests for right now and for the future?
Yeah, so right now I'm working on just finishing my thesis, which following chapters have, it's kind of like a follow-up on this publication that we were talking about. So yeah, I planted seedlings in those disturbed forests, and I'm looking at the associated fungal community composition. Specifically, we're trying to do soil amendments, transferring soils from the control forest into the disturbed forest, just to see if we can somewhat amend the soil community composition, and thus impact or sort of like select for specific ectomycorrhizal fungi that already we know associate with lodgepole pine. So that's kind of like what I'm working on now, as well as doing some tests on whether this ectomycorrhizal communities assemble based on a dispersal mechanism or neutral mechanism, or whether there's some sort of selection by lodgepole pine on specific taxonomic groups that are in the soil. So I'm doing those kind of tests. But yeah, overall, I'm interested in soil microbial communities and the interspecific relationships with plants and other microbes as well. So I'm just currently looking for postdocs in this topic. I'm also interested in like exploring more of the omics technologies. I'm becoming quite interested in metatranscriptomics and see how we can apply these technologies to not only know what the composition of fungi is, but also what's actually the function, because we can make generalizations that ectomycorrhizal fungi does this and saprotrophic fungi does that, but we would actually need to look at what are those transcripts being upregulated or downregulated to see actually what's happening in the system. So I'm trying to like combine multiple omics to have a more clear picture of what's happening.
Cool. Yes. Well, thank you very much for taking the time to talk with us today.
Sure. Thank you.
And I remind our readers that John Carlos is looking for postdocs. So if you have any opportunity, don't hesitate to write to him, or you can write to us as well, and we will put you in contact with him. And we will be back soon with more interview for the South American Mycorrhizal Research Network. Bye.
Thank you. Bye-bye.